Welcome back. Well, the first half of chapter 6, we learned about media, different um, composition of different types of media for growing microbes in the laboratory and culture, forming cultures. And now we're going to look a little more closely at some types of media to start with. Um, Differential media, firstly. Differential media are ones that allow us to distinguish between different types of microbes, usually a couple of different species that we're trying to discern or differentiate between because they're based on their properties. They have a different appearance the way they grow on differential media depending on what microbe it is. In this particular example, we're looking at the ability of microorganisms to lyse red blood cells in the media. So this blood agar actually has blood in it and whole red blood cells. Beta hemolysis refers to a capability to completely lyse the red blood cells. And you can see this completely yellowed area here. The red blood cells have been completely burst open. Alpha hemolysis refers to a partial lysis. You can see some change in color around these colonies, but there's still kind of an orange halo there. Bacteria that do not have the capability of lysing blood cells, that's called gamma hemolysis, which is kind of funny. It means no hemolysis at all. So that's an interesting specialized kind of media for distinguishing between different species of microorganisms. An alpha hemolytic bacteria, Streptococcus pneumoniae, a bacterium that causes pneumonia, as you see in the name, can also cause some other diseases, is alpha hemolytic, whereas Streptococcus pyogenes, which causes a whole bunch of different infections, is beta hemolytic. So that's one way of distinguishing those two from each other. Selective media suppresses some bacteria and allows others to grow. For example, agar that contains a relatively high concentration of salt is selective for staph bacteria, staphylococci, because they're adapted to growing on that super salty environment on the surface of the skin, and they can thrive, or at least survive in that salt medium, whereas most other bacteria can't. And then from there, we can do something else to distinguish different streps or staph species from each other. So, for example, here we have a differential medium, and it's also a selective medium. So it's called mannitol salt. And it has salt in it so that other types of bacteria can't really grow in it, but it has mannitol in it, and Staphylococcus aureus can ferment the mannitol. We can see this yellow color here because they are very avidly fermenting the mannitol and using that as a nutrition source, whereas Staphylococcus epidermidis cannot. And so the medium, the pH indicator has remained neutral, and it's not. We know that this particular culture is not fermenting the mannitol. Here's another kind of selective and differential medium. It's called McConkey agar, and this one has lactose in the medium, and we may determine whether or not an organism can ferment lactose. So here we see lactose being fermented by Salmonella enterica, and the yellow color indicates the acidic products of fermentation, whereas E. coli, Escherichia coli, do not ferment lactose, and so they can grow on this medium, but they can't produce that color change. Also, McConkey agar is a selective medium because it has crystal violet and bile salts in the media, which prevents growth of gram-positive bacteria, so that we are selecting. If we had a big mixture of bacteria, we know that the only thing that would grow on here would be gram-negative bacteria, and then from there we could see which ones can ferment lactose and maybe help us identify. An enrichment culture. Sometimes we're interested in finding out whether or not there's an appreciable amount of a particular bacteria that's very rare in a larger mixture. And so in order to study it or to identify its presence and so forth, we need to get it to become a more prominent member of the mixture. And so enrichment cultures do just that. They allow that rare bacteria that we're interested in to get a foothold and grow, and it suppresses the growth of other kinds of bacteria at the same time. So in this example that's given, a phenol-containing medium can be used to enrich a culture for phenol-degrading bacteria that can utilize phenol. It's a aromatic ring with an OH group sticking out. And so if we grow a mixture of a whole bunch of different bacteria and fungi in this phenol-containing culture, let it grow for a while, and now we have our rare bacteria starting to grow more and more. Take out a sample of that, put it in some fresh phenol medium, and so forth. Do that a few times. Pretty soon we have really allowed the phenol metabolizing bacteria to become prominent in the culture instead of being a super rare presence. So just a quick look at some different types of media we can employ in the laboratory for our benefit. A pure culture, in order to study a particular microorganism and find out what its properties are, we need to get it all by itself. We need to be able to do some tests on bacteria, say a biochemical test, and if there's other bacteria mixed in, we won't know anything about the biochemistry of that particular organism that we're interested in. So a pure culture is one that contains only one species or strain of bacteria and nothing else. So oftentimes when we collect a sample that has this bacteria in it, it's going to have a whole bunch of other things along with it. And how are we going to get that by itself? That's the idea of making a pure culture. What we'd like to do is effectively dilute out all the bacteria in this particular sample, so much so that individual live bacterial cells are separated in the medium to a degree that we can see each live cell will divide and divide and divide and divide and form a colony of identical cells. And then we can pick out one of those colonies and transfer it to some other medium. Now we have a pure culture. We have just picked out a monoculture of bacteria. So that's kind of a fun thing. There's a couple of ways we can do that. A streak plate and a pour plate. Let's take a look at those methods. So in a streak plate, we take a loop, it's called. It's a little tool of the microbiologist, which you've probably already seen in the laboratory. We take some medium that we believe will grow the bacteria of interest, along with other things that are mixed with it. And we take the loop, dip it in the sample, and then streak it back and forth across some part of the agar. And then we turn the plate 90 degrees and then catch the ends of some of those streak patterns and then streak farther. And we're basically diluting things out because we're grabbing a tiny bit of what was the first streak pattern and dragging it out onto a much larger area of the plate. And then finally, we do that a third time, sometimes even a fourth time, and drag just once across all those loops of streak and form a third. And if you look at the results, you can see individual colonies. In this case, probably Serratia marcescens. It's growing in this bright red color. That's one of its hallmarks. And you see some yellow colonies, some other contaminating microbes that were in with it. But we can now, each one of these colonies represents one single colony forming unit, one live cell that was able to divide and divide and divide to form millions, if not billions, of identical bacteria, genetically identical bacteria in these colonies. Now we can carefully reach and pick, mechanically, physically pick that colony out of the plate without touching anything else on the plate and transfer it to some new media. Now we have a pure culture. In the case of a pour plate, we're going to make a series of dilutions of a sample containing the bacteria of interest. So we have a bunch of tubes full of broth. Maybe it's a nutrient broth that has lots of good stuff in there the bacteria can grow on. And we keep on taking out a sample and diluting it 1 to 10. That means that for every mil we take out, we add it to 9 mils in the tube. We wind up with 10 mils total. 1 mil to start with, 10 mils final, 1 to 10 dilution. 10-fold dilution is another way to say that. So we dilute 10 times, 100 times, 1,000 times. And then we take a sample out of each one of the tubes. We may actually go out to 100,000 times. Who knows? It depends how concentrated the sample is. And we just take an aliquot or a set volume out of each one of the tubes and spread it around on an agar plate and then watch what grows. And then to figure out the concentration or the number of colony forming units in the original, we just count the colonies on the plate and then multiply times the dilution factor. If this is 10, 100, 1,000, a 10,000-fold dilution, if we see 4 colonies here, that means there would have been 40,000 colonies, 40,000 colony forming units in the original per mil of the original sample, because again we took 1 mil out and we got the 4 colonies. I'm just making up a number. So that's another way of, we can get a pure culture. I was getting all hung up on being able to calculate the colony forming unit concentration, but also you can see that we pick a plate for making a pure culture. We pick a plate that was um, taken from a dilution sufficient to produce individual colonies arising from individual cells, individual colony forming units. That way we can pick one colony and put it into some fresh media and have a pure culture. Preserving bacterial cultures. Once we want to study a bacteria, when we go to the trouble of making a pure culture, we may want to save some for later because we'll never ever lose it, and we can continue growing the microbe in culture in the laboratory. But we may want to have a something that's more archival, something that will last a long time. And the way to produce that is by freezing at a very, very cold temperature. If we take a broth, for example, of bacteria growing, a liquid, mix it with some glycerol, then we can put it in the minus 70 or minus 80 freezer. Most laboratories have a freezer that's keeping things down at like minus 80 degrees Celsius, which is pretty impressive. And if you stick bacteria that's been mixed with glycerol in that way, it can be kept in there for decades, and the bacteria will survive in there. And whenever you need some, you can go and get some out of the freezer and grow it in some fresh media. Another way to preserve microbes is lyophilization. What you do first is freeze a sample of the bacteria and attach it to a very strong vacuum pump. And the vacuum will suck out water molecules directly from the frozen phase, the solid phase. Sublimation will happen. All the water molecules will eventually all be sucked out, and you'll have a dry concoction, and the bacteria are still able to survive in there. And then you can reconstitute them with some media, and they'll germinate and grow. So lyophilization, freeze drying. Here's a quick introduction to some additional complexity of behavior of bacteria. We said that most bacteria divide by binary fission. They enlarge first, they replicate their genome, and they pinch off and form a wall between two symmetrical parts of the cell, and break into two and have two identical offspring. But there's other ways that some microorganisms can multiply. Here is again binary fission happening. You see that the bacterial cell replicates its genome. I have two identical copies of the chromosome, and then this wall is formed in between, and then finally the two separate entirely, and we have two daughter cells, two identical bacteria. And there's a beautiful transmission electron micrograph of that happening. Budding is something that a lot of yeast cells do, and so instead of forming two symmetrical daughter cells that break off, a small bud, a nub, something will grow out from the surface of the yeast, and then eventually it'll break off in this small, relatively small mini yeast, if you will, containing the genome, will float away and then begin to grow in size and eventually mature to the point where it can undergo budding, replication, or multiplication. Conidiaspores, we'll probably wait till another chapter to talk about of this process, but some bacteria grow in long strands from which pieces can break off that contain the genome and go on to produce more strand-like bacteria, fragmentation of filaments. So we'll talk a lot about complex life cycles of microorganisms that especially fungi, produce all kinds of different reproductive structures, including conidiaspores and so forth. When bacteria grow, the number of bacteria, as long as there's plenty of nutrients, increases exponentially. That means that you can represent the growth in a log way, in a mathematically log scale. And I'll show you what I mean by that in a second. But anyway, think of what's happening with bacteria. They divide by binary fission typically. You start out with one cell and it divides, now you have two, then four. And it just keeps doubling because every one of those cells can then divide and so forth. And it seems like we're not getting very far very fast when we just look at these first few generations. Each time the bacteria divide, that's called one generation. it starts to accumulate. The numbers start to go up very, very quickly, which is a fun, interesting thing about this whole description of bacterial growth. So if we think of going out to a generation number of 20 times, 20 times you've given those cells enough time, enough nutrients to divide, now we're up into a million cells. And it doesn't necessarily take very long to get to 20 generations. If a generation only takes 30 minutes, get to 20 generations relatively quickly, we can go from having one cell to having a million cells, and then that starts to really ramp up after that. So we can grow a lot of bacteria in culture, starting from a pure culture with a relatively small number of cells. The generation time is the time it takes for one cell division to take place for a particular species. So how long does it take after a new daughter cell is formed? How long does it take for that cell to divide again and produce two cells? That's the generation time. There's a cool mathematical thing going on, which I will not ask you to do at any point, but just for fun, you can see that if you know the number of generations and the log number of cells, the number of cells at the end and the number of cells at the beginning, you can figure out from this formula generation time. If that's how many cells we got at X amount of time, then how many minutes must there have been for each generation? And so this math will allow us to figure out, oh, the generation time was, for this particular example, was 21 minutes. But very common rule of thumb for a lot of bacteria that inhabit our GI tract, for example, 30 minutes for a generation time. Some microorganisms, though, like Mycobacterium species, grow very, very slowly. Here's a graphical plot of the number of cells in a culture over here on the right scale. So as they're starting to grow, they're taking a while to get things really rolling. These are the number of generations, and here's the number of cells in parentheses. And eventually it starts to really take off as we keep doubling and doubling. Pretty soon we're getting into some big numbers very quickly. You see the slope of this line, the rate of increase in the number of cells is going up very, very quickly with each additional generation. One way of expressing that mathematically that's a lot more neat and tidy is instead of putting a linear scale down here showing the actually linear scale up here on the y-axis, I misspoke, we can produce what's called a log scale. So if we take the logarithm of each one of these numbers that represents the scale on the y-axis and then plot that way, we'll get a straight line. And it makes it nice and easy to analyze mathematically and to see what's going on there. So that's just a log scale being used to linearize this relationship between generations and bacterial numbers. This is an important graph right here, and it's kind of funny because it looks like they wrote in light, almost invisible colors and drew this line super like dim as though it were almost superficial, but it's actually really, really important. So what you're looking at is a graph of, again, log of the number of bacteria on the y-axis versus time on the x-axis, and we're going to inoculate some broth, say, with some bacteria. Excuse me. And at first, they're just doubling. We have 2, then 4, then 8, then 16. So few bacteria, we can't even see them on our y-axis yet. And at some point, things really begin to take off. We get into that what's called logarithmic growth, that really fast growth in which our log growth forms a straight line on a log scale. So we have this lag phase. It takes a while to get things really rolling. It actually takes a while for the cells to really start even growing, let alone getting a significant number of them. That's the lag phase. And then eventually things take off, and our log scale then looks linear. As long as we have a log scale on the y-axis, we should have a linear relationship between log bacterial number and time, where you could have put generations. And that's when the cells are growing unimpeded. They have plenty of nutrients. There's nothing inhibiting them. They're growing as fast as they can grow. And they're growing by their generation time. Every generation time they double in numbers. Then, after a while, some of the bacteria start to die. And new bacteria are forming by fission, and bacteria are dying also. And when the number of bacteria that are dying equals the number of new bacteria produced by binary fission, we have what's called the stationary phase. The bacteria aren't dead, they're just not increasing in numbers, net numbers, because some are dying off all the time. And so that's what happens when you start to utilize the nutrient. The number of nutrients becomes less available, and more inhibitors, toxic waste products produced by the bacteria begin to accumulate in the medium, and so we start winding up in this situation where there's an equal number of cells dying and being produced in the stationary phase. And then finally, at some point, as the medium is more exhausted and becomes more polluted by waste products, then the death rate is greater than the growth rate. We have logarithmic death. Cells are dying off very, very quickly. Again, this linear slope indicates logarithmic phase of death here of the culture. That's the fate of a typical culture. If you have a finite amount of medium, nutrients, and you inoculate it with some bacteria, you'll have a log phase growth period. They'll enter a stationary growth phase, and eventually the cells will start to die off. So you should be able to explain that graph in entirety, say what's happening at each part of that growth curve of a particular microorganism. So there's a number of ways that we can estimate or actually directly identify the number of bacteria in a particular sample. Some ways of getting at the number of bacteria are called direct methods. We're actually going to do some dilutions and count the results. And we've already been talking about some methods that are like that. I jumped ahead a little bit on that previous slide. But there's some other ways. We're going to use qualitative means to identify the number, to estimate the number, and each one has strengths and weaknesses. So this is called a direct plate count, serial dilutions and a plate count. And it's exactly like we described in the previous slide. We have tenfold dilutions of an original culture, and we're going to take a set volume out of each one of these and spread it on an agar plate, and then count the bacteria at the end, and then multiply the number of bacteria that we get by the dilution factor. In this case, 100,000 is the dilution factor by the time we got to this plate, because we've diluted this one 1 to 10, this one another 10, which is 100, then we diluted another 10, which is 1,000, and so forth. Now we've diluted out 1 to 100,000 times, and we got 3 colonies. So that means that in the original sample there were 300,000 live cells per mil that could give rise to a colony forming units. In order for this to be accurate, we probably wouldn't choose this plate with only 3 colonies on it. We'd choose a plate that has somewhere between 25 and 250 colonies, indicating 250 colony forming units were present, and that gives us a more accurate. People have studied many examples of this and found that this is about the right density to get a pretty accurate estimate what the original sample had in it in terms of numbers of colony forming units per mil of that original sample. You can do this a couple different ways. This exact experiment can be, they're just talking about different ways of spreading out the bacteria on the plate. One way to do it is to add that 1 mil of bacteria from each one of these tubes to some melted agar. It's already been melted at 100 degrees, and then we let it start cooling down. And right before it starts to harden, we add the bacteria to it, swirl it around so it's well mixed, and then let the colonies grow and see how many colonies we get. That's called a pour plate method. Another way to do it is to take an already hardened agar plate and take a set amount, say 100 microliters, 0.1 mils, out of each one of the tubes, place it on a brand new plate, and I don't know if you can see, this is sort of like a triangular shaped spreading tool. It's been pre-sterilized, and we can use it to spread that 100 microliters of fluid all around the surface of this plate and count up the colonies we get. So it's a spread plate method, but it's just ways of getting the bacteria distributed on the plate, no big deal. What if we had a sample? This method works great when you have a lot of bacteria. Look at the bacteria in the sample. It's been diluted 1 to 10, and we still have tens of thousands of colonies on this plate. That's called a lawn. There's not even any way to distinguish individual colonies. It's just overgrown. Whereas sometimes we have a sample that is very dilute. You might take a well water sample, for example. You're the microbiologist. You're sent out to the community where there's suspected contamination of people's wells, of drinking water. And so you might go take a sample of their water, and then you want to see if there's any bacteria in there that shouldn't be in there. Coliform bacteria, fecal bacteria coming from human waste. What are you going to do? Well, there's not very many bacteria in there, if any, and yet that still could be a hazard. So you're going to take a fairly large volume of water and pass it through a filter a filter membrane with microscopic pores so small that even bacteria can't get through, but water molecules can. So maybe you might put a gallon of water through the filter and catch any and all bacteria that are present, and then plate out whatever bacteria were there onto an agar, a nutrient agar plate or something of that nature, or maybe maybe a differential or selective media to identify particular coliform bacteria that might be present in there. So that's just another direct method of actually counting bacteria. We know that we started with maybe 10 liters of fluid. We got this exact number of bacteria. We can calculate how many bacteria per mil must have been initially present. Here's a cool thing. It's called a most probable number method that again has been developed for testing often water samples. And so we might take a water sample and inoculate 5 identical tubes of broth with our water sample. We're going to wind up with 10 mils in each tube every time. The first set of tubes, we might put 5 mils of double concentrated broth and add 5 mils of our test water into there and let those grow overnight in the incubator. You might also take a bunch of tubes with 9 mils in them and add 1 mil of our water sample, and we might take another set of tubes with 10 mils and add 0.1 mils of our water sample, and then count how many tubes have positive cultures in the morning. In this case, we're looking at a fermentation reaction where we see the yellow color indicates the growth of the bacteria that have fermented the sugar in the medium, and we indicate the number of positive tubes. And I guess let's see, I guess I said that backwards. Here are the positive ones, 5 positive tubes. Here are 3 positive tubes in this set of 5, and then only 1 positive tube. And we can go to some statistical tables that have been generated ahead of time, and from the pattern of positive tubes in these sets, we can figure out how many cells were most likely present per mil, live cells in the original sample. So here are some of the patterns of combinations in those 3 sets of tubes, and this is showing us how many cells per 100 mils were probably present in the original. It's kind of an interesting thing. It's just a good method that can handle a pretty broad range of bacterial concentrations and give a fairly accurate estimate very, very quickly. Just set it up, boom, stick it in the incubator, next day, got our number. There is error with it, and here you can see the upper and lower range of the 95% confidence limits. That means we're 95% sure the actual number of bacteria in the original sample would lie between 22 and 150 mils per 100 mils of media, colony forming units per 100 mils of media. But here's our estimate of the actual number here in this column, middle column. Most probable number. Direct microscope counts. In this case, we have what's called a hemocytometer, or something similar. It's a slab of glass, and it's better to look at this drawing. It's a slab of glass with a couple of little wells. See, there's grooves along it, and those grooves are shown here. They're hollow, and there's a mesa, tabletop-like polished surface on the glass that is somewhat lower than the outer shoulders, and we can lay a glass cover slip across the 2 high points, and there'll be a little thin space in between there, and the space in between there is exactly known. And then on the surface of the glass slide is a grating, a microscopic grid of etched lines, so that if we count the number of bacteria within a 2-dimensional grid of known units on each side, and we also know the volume because we know the height of this thickness of the water that would fit, or the media that would fit in between the slide and the cover slip, we can figure out number of cells per mil by multiplying times some dilution factor, or the relationship between the tiny volume in one of these squares versus a larger number like a mil. So in this case, we're going to look on this like in the microscope, and here's what we're going to kind of see. Let's say we're going to count the number of bacteria in one of these large squares right here that I'm outlining. Might count them all up and count maybe 5 different large squares and write them down and take the average. And then we might then, to find out how many cells per mil we originally had, we might multiply that by some value, however many, whatever fraction of a mil each one of these little squares represents, might multiply it times 50,000 or whatever to get back to our original. In this case, they're saying that the volume of fluid over one large square is 1,250,000th of a milliliter. So we're going to multiply 14 times 1,250,000. And our original sample would have had 17,500,000 cells per mil if this is what we saw in the slide. So you get the idea. You count the number of cells in a known tiny volume, multiply back, and know how many cells per mil you started with. So it's just... Now we get into an indirect method. Those are all direct methods. We're actually counting the bacteria. We're counting the colonies that came from colony-forming units, live bacteria that could multiply. And before we go on, actually, I want to back up one step and just say one more thing to you. If we use a method like this direct microscopic count, we can find out how many bacterial cells there are per mil of sample. What we don't know is, are those live or dead cells? So that's a disadvantage. The advantage is this is extremely quick. We can do this in a few minutes time, get our sample, walk over to the microscope, put the sample on this specialized slide, count, and we're done. The problem is we don't really know how many live cells there are, whereas if we go back to a method like this, or like this, or like this, we are only looking at cells that were colony-forming units that were able to grow into a colony and produce a result that way. So there's an added advantage to using a method that involves culturing the cells, but then we have to wait overnight for the cells to grow. Here's an indirect method. You can grow bacteria in some broth medium, some liquid medium. You first inoculate some broth with a tiny bit of bacteria, and then shine a light through there on a spectrophotometer at, say, 600 nanometers wavelength, and we'll get a reading for how much light would be transmitted through that media in the test tube. And then as the bacteria begin to grow, more and more bacteria begin to scatter the light, and less light comes to the light sensor, the photomultiplier, and at some point we'll have a high density of bacteria, and the absorbance or the transmission of bacteria, the light transmission is going to go down and down and down. And from that drop in light transmission, we can make some estimate of the number of cells of bacteria, the concentration of bacteria in this sample here, and then from that we can figure out how many must have been in the original sample by knowing the generation time. So it's just a more qualitative way of seeing how many bacteria we think was in the original sample. Again, we're not counting anything. We're just looking at a property of a large number of bacteria in solution to be able to scatter light, and giving us a rough way of estimating how many bacteria are in that suspension, how much were in the original sample. There's other ways that are indirect. That was called a turbidity measurement. A turbid fluid is one that has a lot of stuff suspended in it. It's not clear. Another way to do it is measuring metabolic activity. What if we went and measured how much glucose was consumed per minute by a culture of bacteria? There's ways of estimating from that kind of data how many bacterial cells must be present in the media. Another way to do it is to grow the cells for a certain number of generations and then dry the whole thing down. Get rid of all the water, and we're left with nothing but the bacteria, the mass of the bacteria. And from that final dry mass or dry weight of the bacteria, we can figure out how many cells that represents because of many, many experiments that have been done to establish that connection. So some direct methods and some indirect methods to estimate how many bacteria are present. All right, well, that wraps up Chapter 6, and I hope you enjoyed that. Next time we'll meet up again to go on for next week's series of chapters, which begins with Chapter 7.